Hello, my name is Alban, and I am here to present our poster on single-cell deconvolution for spatial transcriptomics in cardiorenal disease. The results shown here were created in a collaborative project between Novo Nordisk and the Technical University of Denmark. A central limitation of spatial technologies, including the 10x genomics Visium solution used here, is that the resolution is not at the single-cell level, and even for high-resolution methods, the spatial unit or pixel is not guaranteed to align with each individual cell. And so transcriptomic profiles will stem from a mixture of a small number of cells and often more than one cell type. Addressing this challenge, a recent search in computational methods have been published with the goal of deconvoluting the spatial signal into constituent cell types with the use of single-cell transcriptomic cell type profiles. Recently, benchmarks of such methods have shown promising performance in various tissues, such as mouse brain, other mouse tissues, and human cancers. We aim here to validate the most promising of these methods in normal and pathological samples from human arterial blood vessels and kidney samples relating to cardiorenal disease. For this project, the arterial cohort of 10 spatial samples were generated at Novo Nordisk using the Visium solution. 5 samples contained arterial plaques, 3 were healthy, and 2 in the normal range. A reference with cell type labels was assembled from multiple publicly available single-cell RNA-seq datasets. For arterial samples, the main cell types present were smooth muscle cells and smooth muscle cell hybrids, endothelial cells, fibroblasts, macrophages, as well as other uncommon immune cells. Based on existing benchmarks, the 3 packages Robust Cell Type Decomposition, or RCTD, Spatial DWLS, and Cell-to-Location were selected, and each method was used for deconvolution of the 10 samples. Each package works in a supervised manner using the single-cell RNA-seq data to build cell type expression profiles before decomposing the spatial data. Both the weighted least squares approach of spatial DWLS and the probabilistic method of RCTD can deconvolute a spatial sample in a few minutes. The Bayesian model cell-to-location is more computationally demanding and relies on a deep learning framework, which in turn provides a more customizable and a probabilistically detailed model. A partial ground truth for cell types was obtained from an experienced histologist with tissue-specific expertise at Novo Nordisk Pathology Department. Using this to benchmark, all 3 methods perform well in predicting dominant cell types, macrophages, and smooth muscle cells. In contrast, the performance was generally poor for endothelial cells located in the lining of the arterial lumen. Performance was evaluated using an accuracy score for these verifiable cell types, resulting in similar scores for all 3 methods. RCTD did achieve a slightly higher score for arterial samples at 0.73. Generally, the level of agreement between the 3 methods was seen to be higher for major cell types and less so for uncommon cell types, for which exact ground truth validation remains a challenge. When repeating our analysis multiple times with randomly subsampled reference data, cell-to-location was found to arrive at the same cell type decomposition with less reference data available. With as little as 100 instances randomly sampled from each cell type in the reference data, cell-to-location achieves more than 95% consistency, whereas DWLS shows less consistency for uncommon cell types, even with larger data samples. All 3 methods perform well in demarcating tissue regions, including plaque lesion morphology, as validated by expert histologists. Similar results were achieved when deconvoluting a smaller cohort of human kidney samples, including healthy and pathological chronic kidney disease samples. Of particular interest here were the rare but easily locatable podocytes of the glomerulus, which were predicted with very high accuracy by all three methods. I hope this has helped elucidate the current state and potential of these methods for maximizing insights available from spatial methods. If you have any questions, feel free to reach out.